Very good morning, students. Today we will be studying a new topic, that is karyotyping. Now when you talk about karyotyping, karyo means nucleus. Typing in the sense, inside the nucleus we have got 46 number of chromosomes. So during karyological studies, or when we make this karyotyping, inside this nucleus, we are completely removing this 46 number of chromosomes, and these chromosomes are arranged in the form of a diagram. So this arrangement of this chromosome, so in karyological studies or in karyotyping, inside the nucleus, cell will have a nucleus. Inside this nucleus, you will have 46 number of chromosomes. In this karyological studies, we are removing this entire set of chromosomes from the nucleus and they are arranged in the form of diagrams. And this arrangement of this chromosome in the form of a diagram is based on the size of the chromosome, the shape of the chromosome, as well as the banding pattern. The banding pattern means we are talking about the two bands, that is, one is the dark band and the other one is the light band. So we will talk this banding pattern in detail a little later. So now in karyological studies or in karyotyping, inside the nucleus we have 46 number of chromosomes. So we are removing this 46 number of chromosomes, and all these 46 number of chromosomes are arranged in the form of a diagram on the basis of the size, shape, as well as the banding patterns. And since we represent these chromosomes in the form of a diagram, this is also called as ideogram. So karyotyping is also called as ideogram. These are also an important one-mark question. Now, why we are doing this karyotyping? What is the necessity of removing this 46 number of chromosomes, and based on these size, shape and banding pattern, why we want to organize or arrange in the form of a diagram? And that is because in order to find out any genetic disease, we have to examine the chromosomes, the shape of the chromosome, the size of the chromosome, and based on the banding pattern, we have to examine the genetic chromosomes so that we can easily identify any genetic diseases. And that is the reason we are going for this karyotyping. Clear? Right. So now, these 46 chromosomes, how they are organized, that is, this arrangement is always done under the microscope. Why? Because viewing the cell with our naked eye is not possible. Viewing the nucleus is also not possible. But how we will examine the chromosome? We cannot view the cell, we cannot view the nucleus, but we have to examine the chromosomes, entire set of chromosomes which is located in one particular cell, in order to find out any genetic disease. So karyotyping will help us to see the chromosomes clearly by certain methods that we will discuss now. Now, so what we are so far discussing is, we are talking about karyotyping. In karyotyping, inside the nucleus we have 46 number of chromosomes. Half comes from your father and other half comes from your mother, and these 46 number of chromosomes are arranged in the form of a diagram based on the size, shape or the banding pattern, that is dark band and the light band. And this karyotyping is also called as ideogram, which means diagrammatic representation of the chromosomes. So chromosomes are represented in the form of the diagram. So that is what the ideogram means. So next one, here these 46 number of chromosomes are arranged in 7 different groups. So the diagrams will be there. Afterwards we will concentrate the diagrams. So now this entire 46 number of chromosomes are arranged in 7 groups. So that is from A to G. So that means A, B, C, D, E, F and G. And I'll give you a number for remembering the number of the chromosome which is located in each group is, that is 3, 2, 7, double 3 and double 2. So this is the number of the chromosomes that is we have. So if the group is A, it will have 3 number of chromosomes. For example, chromosome number 1, chromosome number 2 and 3 comes under the group called A group. Then 4 and 5 is B. Then 6, 7, 8, 9, 10, 11 and 12. Those chromosomes comes under the group called C. Okay. And 3 here. So we will have 13, 14 and 15. Then here we will have 16, 17 and 18. Then 2, 19, 20 and here we will have 21 and 22. So up to 22 we will have group G, and the another 2 chromosomes are sex chromosomes, that is comes under the 23rd class. All these are pair homologous chromosomes. Okay. So here we will have the X chromosome and Y chromosome. Always remember the X chromosomes comes under the group called C group. So wherever 6 is there, remember X chromosome is also there for female. And wherever G is there, you will have Y chromosomes. So now if you can remember this number and this group, you can easily note down the number of chromosomes. See, you will have A to G, 7 groups are there. The number is 3, 2, 7, double 3, double 2. So that easily you can place the number of chromosomes in a particular group. Clear? And here, the largest number of chromosomes are located in group number C, as well as the large chromosomes are presented in the group number A. So that is what we are organized on. Based on this, we are arranging the number of chromosomes in the form of an ideogram, or it is diagrammatic representation of the chromosomes. Clear? I'll just come back once again. So far we are discussing about the karyotyping. Karyotyping is done for examining the chromosome in order to find out any genetic disease or disorders. So how we do this karyotyping is, inside the nucleus we have 46 number of chromosomes. So we remove this 46 number of chromosomes, and under the microscope, based on the size, shape and the banding pattern, the chromosomes are arranged in the form of a diagram under 7 groups. Based on this number, the chromosomes are represented in the form of a diagram. So we have this called ideogram. Clear? Now we will talk about how this technique is being done. How this technique is being done is, Theo Levin in 1960, they have described a very simple method of culturing the lymphocytes. The lymphocytes are the one type of WBC that we will have it in the blood groups. So what they are in blood, so what they have done is, they have cultured this lymphocyte. So how they have cultured this lymphocyte is, for example, and this is the lymphocyte of the human being. So now they have taken this lymphocyte from a human and they have cultured this lymphocytes for 3 solid days in the laboratory. Okay. So in the medium, in the nutrition medium, they have cultured this. Now, these lymphocytes, now if this is present inside, this is the actual lymphocyte which is having a kind of nucleus here like this. So now the chromosome which is present inside will be in the form of a condensed thread-like structure. So this thread-like structure is called chromatic reticulum. So this is what the structure of the chromosome which is present inside the nucleus. So this cell is the lymphocyte, where Theo and Levin in 1960 they have cultured this lymphocyte from the human blood. They have taken this lymphocyte and they have cultured this for 3 days in the laboratory in the nutrition medium, and inside this nucleus we have called this chromatic reticulum. And now this cell, when they culture, the cell will start to divide. So whenever the cell is start to divide, at the time of cell division, this chromatic reticulum will be, this is a chromatic reticulum, will be forming a kind of a division and it forms the chromosomes. So like this, the chromosomes are formed. So this chromatic reticulum forms the chromosome only at the time of cell division. As the cell divides, when the cell divides, this chromatic reticulum which is kept inside the nucleus will start forming the chromosomes. Okay, so now this kind of a cell division which is taking place is called mitotic cell division. Very important. Now, so that means karyotypical studies or karyotyping, or if you want to study the size, shape of the chromosome, we have to go for only mitotic cell division. So karyotypical studies are carried out in the mitotic cell division only in the body cells, not in the reproductive cells. So you can do it in the meiotics, but chromosomes detailed can be best studied only in the mitotic. Though you can do it in the meiosis, but taking body cell is very easy comparing to the reproductive cells, and that is why most of the karyotypical studies are made in the mitotic than in the meiosis, because it's easy to obtain this somatic cells, body cells. Clear? Right. So Tejo and Lehman, they have isolated this, they made cells lymphocyte from the human blood and they cultured in the laboratory for 3 days. By the time this chromatic reticulum will undergo mitotic cell division so that it forms the chromosomes. Clear? Right. So now this whenever the cell divides, we have certain phases or stages in the cell division that we already discussed in the 11th standard. That is, the cell division is when we have 4 different stages like prophase, anaphase, metaphase, telophase, all this. Now here, whenever the cell division reaches the metaphase, the cell division has to stop in the laboratory. Why? Because the chromosomes can be clearly seen only in the metaphase. Why mitotic? Obtaining the cell is easy. Why metaphase? Because the chromosomes are visible clearly only in the metaphase. And after the spindle fiber forms and this chromatin, all this chromatin fibers, everything will be separated. So the ideal stage or the ideal phase where you can clearly study the shape and the structure of the chromosome is in the metaphase. So what should happen is, during this cultural process, the cell division should stop at metaphase. So what we are doing is, we are using a chemical to stop this cell division at metaphase is called colchicine. A very important chemical component that stops mitotics at metaphase. So we are having a chemical called colchicine. Now when we add this chemical colchicine to this cell which is being cultured, so here we are going to add this chemical called colchicine, and the colchicine, when the cell is undergoing division, mitotic division, when the time of metaphase, it will arrest the cell division. So this colchicine is also called as mitotic inhibitor. Mitotic inhibitor in the sense it will stop the mitotic cell division at the metaphase. Okay, so why cell division is stopped at the metaphase? Only in the metaphase you can see the chromosome structure, size, shape, everything clearly. Okay, so that then only, so how you can view this chromosome in the metaphase? Under the microscope. So by viewing this microscope, under the microscope, the chromosome structures, shape, everything will be clearly shown when the cell is stopping at the metaphase. Okay, so now these cells are arrested in the metaphase by using a chemical called colchicine, that is also called as mitotic inhibitors. Okay, then after this, if you add a kind of a dye in the sense a stain. So whenever you add a dye, now for this cell division, we are using a particular dye. So that particular dye, when that dye name, everything is not given in the book, but still you know. You learn that is. So now we use a particular dye called Giemsa dye. The nature of this Giemsa dye is, in the the DNA, okay, in the DNA. So with the inside this chromosome we have that DNA. So everywhere we have these genes, everything. So wherever 80 rich region is there, wherever 80 rich region is there, this 80 rich region will absorb this dye and produces a dark band. So wherever 80 rich region is there, this gypsa dye will go and add to this, and wherever 80 region is there, it becomes dark, and wherever G and C region is there, it becomes very light. So this region is the light region and this region is the dark region. So out of 46 number of chromosomes, okay, out of 46 number of chromosomes, we have done this karyotyping. And end of this karyotyping, we have added a dye to this. Now, suppose if this is a first number of chromosome and the dye is here 1, 2, 3, you have to find out same kind of chromosome among this 46. One is already there. So, out of another 45, you have to find out same kind of a banding, thrice same kind of a banding is done among this 45 and you have to pick it up. Okay, so now before this doing this procedure, after all this dyeing everything, you have to place this entire set of chromosomes under a slide, keep under a microscope, take a photograph, and in the photo you will have all this 46 number of chromosomes. So now you are cutting with a scissor particular number of chromosomes, that is having a dye, that is thrice. Same kind of a chromosome if you find in this group, you have to isolate that and place based on one group. So now this is the one pair of chromosome which is homologous. Homologous means same kind of banding is there. That is what in the beginning itself we studied. Karyological studies or karyotyping is done based on the size, shape and the banding pattern. So now these bandings are more or less both are similar. So like this, you have to pick it up all this 46 number of chromosomes and arrange the pairs. So that means one comes from your father and one comes from your mother. So totally you will have a pair of chromosomes. So this karyological studies are done for entire 46 number of chromosomes. So what we have done is Theo and Lehman in 1960, they have isolated the lymphocyte from the blood cell of a man and they cultured the laboratory for 3 days along with the adding a chemical called colchicine, and this cell undergoes mitotic division. By the time the chromatic reticulum will start dividing and forming the chromosomes, and this colchicine, as the cell is undergoing mitotic cell division, it will stop mitosis at metaphase. So this colchicine is also called as mitotic inhibitor, where it arrests the cell division at metaphase. Why? Because chromosome structures can be clearly seen only in the metaphase. And after that we add a dye called gypsa dye, and the gypsa dye will be added wherever 80 region is there, it will go and makes the dye of it, so that all the 80 region will become dark band and wherever CG region is there, it becomes light. Now keeping all this entire process under, keeping this entire cell under the microscope and take a photograph of it, and in the photo you are identifying the same homologous chromosomes out of this 46 and cutting those individual chromosomes and placing in pairs, so that you can place all this 23 pair of chromosomes based on 7 groups. That is what we studied from A to G. So the number is 3, 2, 7, 3, 2, 2 and X chromosome and Y chromosome. Now by seeing this picture of the chromosome, easily you can identify whether if any defect or in the size or shape or in the banding pattern, if anything is defect, you can easily identify the genetic disease of a fetus, of an young one. And that is the reason we are going for the karyotyping. I hope you understand this. And the applications are your book bag question in karyotyping. The applications are very, very important. This for the main aim of doing karyotyping is to identify the genetic disorder or genetic disease. But you can even identify you can easily identify the genders of a fetus. So easily you can identify the gender. So it will help to identify the gender. For example, if a male or female can be easily identified by doing the karyotyping studies. And any chromosome abnormalities, can be easily identified by this. Chromosome abnormalities means, otherwise it is called aneuploidy. Aneuploidy means when the cell is dividing, the chromosomes are not equally shared. Maybe one less number of chromosomes or more than one chromosome. One chromosome is added more. So that is called aneuploidy. So that means aneuploidy is either loss or gain of the chromosomes. That is what aneuploidy is. So this karyotyping is not only doing for identifying any genetic disorder, but still we can identify the gender, and any chromosome abnormality such as aneuploidy can be detected. In the sense, any gain or loss of chromosome can be easily find out by this karyotyping. Clear? Right. So that is what karyotyping is. So in karyotyping, we have concentrated that this is a complete set of chromosomes are separated from the cell, number one, and it is also called as ideogram, which means diagrammatic representation of the chromosomes. And who has first described this is Fijo and Lehman in 1960 with lymphocytes of human. What they have done, they have cultured this cell for 3 days in the laboratory under optimum temperature by using a chemical called pulsicin, which is called mitotic inhibitor, that will be stops mitotic cell division at metaphase. So two things we have discussed about this mitotic and metaphases. Mitotic, the cell can easily obtain. Metaphase, it will stop cell division at metaphase so that chromosomes are clearly visible under metaphase. Okay, and adding all this dye, so based on the banding pattern, the chromosomes are being arranged. So all the homologous pair of chromosomes are arranged in the following 7 groups that we have already discussed. So that's all about your karyotyping. One part that we will discuss now, that is the type of the chromosomes. Actually, chromosomes are divided into 3 main types. So now this classification of this chromosome under 3 types is based on 2 important things. One is the position of the centromere. You know what is centromere? Now this is the arm of the chromosomes. This is the arm of the chromosomes, and this arm is placed on the centromere. So now this is the centromere. Okay, so this is what the centromere is. Now, so this one is the arm of the chromosomes, arms. So now this classification of this chromosome, that is 3 types, this classification is based on the position of the centromere, where the centromere is located. Second, the length of the arm. So based on this, the chromosomes are being classified into 3 types. One is based on the position of the centromere and the length of the arms of the chromosomes. Okay, so now the first type is called the metacentric, or otherwise called as metachromosome. So metacentric. So now it is metacentric in the sense the centromere is positioned in the center, and the 2 arms are equal. So see, this one arm and this is another arm. So this is one and this is one. So the arms are equal. So that is what metacentric are. This is the first chromosome. So chromosome, 3 classifications based on the position of the centromere and the length of the arm. The centromere is positioned in the center and the 2 arms are equal. So it is called metacentric. The second one is submetacentric. Submetacentric. Now submetacentric in the sense the 2 arms are unequal. these arms are lengthier and these arms are short. So it is, the arms are unequal. So this is the second classification of the chromosome. First one is metacentric, second one is submetacentric, and the third one is agrocentric. This is the third one called agrocentric. Now, agrocentric chromosomes, the top you will find like this, and the below you will find one more like this. So this is what the agrocentric is. Clear? Right. So I can even put 2. So but still you can understand. So you will have agrocentric means here the agrosome, the top will be having very short, and you will have the lengthy one, the base. And one more type that we will have that is not discussed in the book, that is called telocentric. The fourth one is the telocentric. In telocentric, we will not have any arm on the top. Only below this you will have these 2 arms. So this is called the telocentric. So there are 4 types. Actually, there are 4 types of chromosomes, but we have discussed only 3 main types. That is called metacentric, 2 equal arms. Second one is submetacentric, that is 2 unequal, and one is called the agrocentric. The top will be little less comparing to the other part of it. And the last one is the telocentric. And that's all about your karyotyping. Now the next topic that we will discuss now is pedigree analysis. In pedigree analysis, a very important statement that you have to understand is, it is inheritance pathway for a specific phenotypic characters. Inheritance pathway for a specific phenotype characters. You know what is inheritance? A character that is being passing from one generation to another generation. For example, a character that is passed from your grandparent to the grandson. So that is what the inheritance are. Any character that is being passing from your grandparent to the grandson is called inheritance. But here what we are focusing is, this inheritance pathway speaks about a specific phenotypic characters. What is phenotype? Morphological characters. So a particular morphological character, an external appearance, is being transferred or inherited from your grandfather to the grandson, and that is what inheritance pathway for a specific phenotypic character is. Here, if a particular character is being passing from your grandparent to grandson, is only through the genes. If the gene is normal, the gene will be passing to the grandchildren, and the grandchildren also will be normal. Suppose if this gene is abnormal, this abnormal gene will pass to the grandchildren, so that the grandchildren will be affected with this abnormal gene. Okay. So here in this pedigree analysis, we are mostly focusing on the abnormal gene that is going to be there. So here we are focusing on the abnormal gene which is being inherited from your grandparent to the grandson. Okay, so that is what pedigree analysis is. So here, suppose if the gene is abnormal, that abnormality can be identified by using this pedigree chart. Here, the pedigree chart gives you a complete history of your family. That is why the pedigree analysis is also called as your family tree, because it gives you a complete picture of the entire families. If the gene is abnormal, by looking at this pedigree chart, by doing this pedigree analysis and studying the pedigree chart, easily you can identify that abnormal gene has come from whom and to whom it has been sent. So who has got that gene, abnormal gene, to whom it has sent, who has received it. So everything crystal clear you can study by analyzing the pedigree analysis. Clear? So I hope you understand what this pedigree analysis is. Here, a particular specific phenotypic character that is being inherited from a grandparent to the grandson. Suppose if the gene is normal, no problem, the grandchildren will have normal characteristics. Suppose if the gene is abnormal, that abnormal gene will definitely pass to the grandchildren, and later on the grandchildren will be affected with certain hereditary diseases, certain genetic diseases. So here, this pedigree analysis gives you a complete picture of that particular family who is being affected with a normal gene, and it tells you from whom that gene has come and who is being affected and all this. Now here, this in pedigree analysis, we have a certain standardized symbol for certain things. Okay, so now this, most of the time, some application oriented question, need based questions will be asked based on this. First you learn what is this pedigree analysis, and later on we will be discussed during our revision time, we will be discussing about certain application oriented questions based on this pedigree analysis. Clear? Right. So now here we have some standardized symbols in the pedigree chart that we have to learn. That is what, only this much is given in your syllabus. So we learn what is pedigree analysis. In pedigree analysis, inheritance, it is a inheritance pathway for a specific phenotypic character, and it is drawn by a standardized symbol, and this is also called as family tree. Why family tree? Because it gives you all the complete picture of a particular family. So it is called the family tree. Now here we have certain symbols I have drawn here for you to understand. Now if you find this kind of a square, and this is represented as male. So if you see this kind of a thing in the pedigree chart, this always represents male, and this symbol is for female. Okay, and if you find one square and one circle with this connected with a small dotted line, and this is the mating, that is the parent. So mother and the father. Okay, and here this parent has got two children. This is the children, and this will have one male will be there and one female will be there. So this is male and this is female. Okay, and this symbol denotes that if it is having one male and one female with this symbol, it indicates that is the dizygous twins. Dizygous twins. So this is what very important symbol that we have to understand. Certain times it is asked. So this one is the like if you have the symbol like this, so it is called dizygous twins. If you find this both, so dizygous twins, both sex will be different. Though they are called twins, but the sex both will be different. One male and one female. And this one is the monozygous twins. See, monozygous twins means both the sex are same. Both will be male or both will be female. Okay. Now, if you find the symbol like this is called the sex which is unspecified. It is not correctly specified. So, the sex which is unspecified. So, that is what the symbol which is given here, the sex is unspecified. Okay. And if you find this kind of a symbol which is fully shaded, and this is called the affected. These individuals are affected with certain abnormal diseases or abnormal genes. So definitely they are, if you find a symbol like this which is being shaded, both male as well as female are affected with certain genetic disorders. Okay, and if you find one part is shade, half it is shade here for male and female, they are called carriers. Why we call the carriers? Actually, you know that for them it is not a disease, but they can carry the disease to the another generation. Okay, so here we have the heterozygous or autosomal recessive. So this is called heterozygous. Heterozygous means only one part, one gene will be affected. Okay, heterozygous. Out of two alleles, one allele becomes affected. So how this allele? The allele is heterozygous. Okay. By the time, though it is heterozygous, means that both are not same. Heterozygous. For example, capital A, small a. So this is called heterozygous. And this is also called as heterozygous autosomal recessive. It is called autosomal recessive. Recessive, the autosome is body chromosomes. Recessive in the sense both chromosomes become small. Small a. For example, if mother is like this, mother's gene is like this, and father is also having same. Okay, heterozygous. So now one children not affected. This is carrier and this is also carrier. So here you will find small, both A are recessive. So if this condition is expressed, that is called heterozygous autosomal recessive characters. If you find these kind of characters or this kind of both recessive alleles are there, definitely they are going to be the carriers of that disease. So they are going to spread this disease from one generation to another generation. That is what it is mean heterozygous for autosomal recessive. Clear? Right. Now, if you find this kind of a structure which is for female, so that is the same thing that is happening for the is a carrier for sex linked. This is also a carrier, but here we have this is only female are carried in the sex linked chromosomes. So it is called sex linked recessive. Okay, see, in autosome heterozygous recessive, both can be carried, male as well as female can be carried, autosomes. Most of the time, the carriers are being expressed or passing from one generation to another generation through these sex chromosomes only, not the body chromosomes. Okay, they are not having the disease, but still they are the carriers. Okay, so that means they have the heterozygous condition. In heterozygous condition, those who are having heterozygous like this, these are called carriers. If you find this is carrier and this is also carrier and this is normal because both are capital. But here if it is having 2A, it is in recessive condition, both A are recessive, that is called as the disease. Okay, but here we don't find this kind of a homozygous, but here we have only the recessive. This is also capital A and small a, capital A and small a. So this is heterozygous autosome recessive. So it is not going to spread to another generation because it is an autosome, body chromosome. But whereas if you find sex chromosome, sex linked recessive, that is only for the female. Female are the carriers. They are called sex linked recessive and they become the carriers. So now, these female when they become carrier in the sex chromosome, for them it is not a disease, but definitely they are going to spread this disease to the young generation. So the difference between this heterozygous autosomal recessive and sex-linked recessive is in autosomal recessive, both are heterozygous, but still they are not going to spread the disease. They had the chance of having the disease. That's it. But when you have sex-linked recessive, this female is going to cause this disease or spread the disease to the young ones. Okay. And the last one, if you find a symbol like this, there is a death in the family. That is what the indication of this symbols are. Okay. So far we are discussing about the pedigree analysis. The pedigree analysis will give you a complete picture of the inheritance pathway for a specific phenotypic character in a particular family. That means if a normal gene is passing from the grandparent to the grandchildren, they will receive always a normal gene. Suppose an abnormal gene is being passed to the grandchildren, by looking at this pedigree chart, by analyzing the pedigree chart, you can easily identify who is responsible for passing that abnormal gene to that young generation or to the grandchildren. Okay, so this is being drawn by certain standardized symbols, and that is what we have so far explained. Clear? Right. So today we have discussed two different topics. One is called karyotyping, and another one is called pedigree analysis. And the next following classes will be discussing about the genetic disorder, and with that we will successfully complete our fourth chapter. Okay.